and it's my pleasure to introduce Cara Santelli. Cara is an associate professor in this ESI department, Earth and Environmental Sciences, and also the BioTechnology Institute. Cara got her PhD in the MIT Hui Joint Program, and then she went on to do a postdoc at Harvard before being the curator of minerals and gems and a researcher at the Smithsonian Institution. Cara is a recipient of a NSF Career Award and soon to be the recipient of a GSA Geomicrobiology Section Award. So congratulations on that. That's exciting. Cara's lab investigates the impact of microbial processes on a wide range of different geological and environmental processes. And in addition to answering key questions of mechanisms, metabolic pathways, geochemical impact of micro-mineral interactions, her research seeks to inform and improve strategies for remediating pollutants to improve the health of water and soil environments. Her work also seeks to advance environmental justice through community-based participatory research that prioritizes and addresses the voices of communities. Cara also started and for many years chaired the ESI DEI committee, as well as the College of Science and Engineering Diversity and Inclusivity Alliance, and really pioneered a lot of the DEI work that's going on in this department and the college and university broadly. So really, Cara wears many hats and does lots of important work in many fields. And so today, Cara is going to be telling us about sulfur cycling and hydrobiogeochemistry.
All right. Oh boy. Okay. No, this is going to be hard to keep. Can you hear me? I'm going to try my best to, I don't know. This is a floppy sweater. I should have thought about that. Okay. Well, it is so nice to see people. This is the reason I'm giving a talk today. I gave a talk maybe 3 years ago for my tenure talk, and now I'm asked to give another one in the department is because I took a sabbatical last year. So it means I haven't seen people face to face in literally like 2.5, almost 3 years. So I'm so happy to be like seeing people again and giving a talk in person. It's really nice. And hi to all the folks online as well. I appreciate it. So I'm going to just give an update. I'm going to talk about this project, of course, about sulfur cycling that we've been working on for a few years, but I'm also wanted to start with, an introduction to my group, because I haven't seen a lot of people. I don't even know a lot of the new graduate students, new researchers in the department, because I was gone last year. And then COVID, as we know, I don't have to describe COVID, but okay, let's see if I can go forward. There we go. Okay, so when I first started out here at the University of Minnesota, my lab group was really, I considered myself a geomicrobiologist, so microbes interacting with the environment. And that's still very much the focus of my research group. But I've expanded a lot over the years, and I've kind of spent some time when I was preparing this lecture thinking about, how my lab group has evolved over the years, which is what you want to see in terms of growth as a professor throughout their career. And I thought a lot about what do we call our group? And so, it's kind of expanded to this more environmentally focused and applied biogeochemistry group. And we still have, microbes at the very bottom, the smallest things, as well as, the precipitation of biominerals, interactions with metals. That's how I started out. And it's very much at the core of my research program, right? But I started to, throughout my career, collaborate with new people here in the department, throughout the university and beyond, that has taken my research program in newer directions that are sort of much bigger in scope and in size, right? I used to be really focused on sort of molecular scale interactions. And now we're starting to think about, okay, well, these molecular scale interactions are so important for the environment, but how do we start to think about scaling that up, right? How do we take what's happening with an individual microbe or nanoscale minerals in the environment and scale that up to think about the entire environment? So that's a lot of what my research has been trending towards in recent years, is thinking about this next scale, okay? And to do that, we do a lot of field observations, but I've started working with a lot of modelers to try to scale up to say, thinking about some of these processes at the small scale, how they impact polluted environments, pristine environments, maybe a single river or a single lake, and how do we scale that up to larger scale, thinking about entire watersheds. Okay? And so that's a lot of what our research focuses on. But as part of this, we're also starting to think about societal impact. Of course, I've always been concerned about the environment and how this work impacts society, but my work never really incorporated humans in it, right? We never really interacted with the community. We never thought really clearly about how human society was impacting the environment in subtle ways. And so now we're starting to incorporate and working with people like social scientists and others, and working directly with communities to think about the community impact on these environments and vice versa. So that's sort of where my group is today. I wanted to give a picture of all of my group members. These are the people that bring me joy. We talked Monday in my lab group meeting about how my last sabbatical year was kind of focused on reminding myself what brings me joy. And I have a lot of joy in my personal life, and I have a lot of joy in my professional life, but during the pandemic and the sort of the intensity of trying to get tenure, you start to, at least I started to lose a little bit of that. And so I wanted to go back to what brings me joy. And these are the people and their science and what they do for each other, what they do for their science, what they do for their communities and beyond. These are the people that bring me an incredible amount of joy. All of my grad students here and postdocs in the group, some of the faces you might know: Chris Schuler, Amanda, Sam, Shreya's a postdoc who's also here. Christoph is Christoph is a new grad student in the group. Brandy Stewart, Sarah Dance, and Paula B's a new postdoc in my group as well. So I just wanted to highlight them because they're the people doing all of this work that I'm going to be talking about today. There's a lot of different projects that they're working on, but they, I just wanted to acknowledge them because they are just like really an amazing group of people. And they got me through some really tough times during COVID. So I'm focused on them and the joy that they bring. Also, other things that bring me joy: yellow shoes. I'm sorry, but I am focusing this year on what brings me joy in my professional world. And so therefore, I will be wearing yellow shoes a lot of the time. Okay, anyways, but that aside, so today I'm going to be talking about riparian wetland hydrobiogeochemistry. I decided biogeochemistry wasn't just enough. I had to add the hydrobiogeochemistry. And so this is a collaboration that's been going on for a number of years, a lot with Crystal Ng, one of our hydrogeologists in the department, many of you know. And what we're focused on is understanding sort of the dynamic interactions between geochemistry, the microbes, as well as the physical processes like hydrogeology, understanding all the connections between them, and how this impacts things like bioavailability of nutrients, of carbon, flux of contaminants, for example, in streams and wetlands in the hyporheic zone. So with that, I just want to briefly introduce, we have a couple different projects going on in the lab, but what I'm going to talk about today is a project in collaboration I mentioned with Crystal, but with a number of other people at Argonne National Lab, as well as a few other places. And these are the people who generated all of the data that I'm going to present today. Shreya has done a lot of the microbial work that you're going to see. Carla is a collaborator. She was a former postdoc of mine and now a researcher, and I work a lot with her. Crystal and Sam and I. Sam is a graduate student working with Crystal and I on the reactive transport modeling, and I won't get too much into that today. And Aubrey was a master's student here who really kind of helped us spearhead all of this. So she was an important part of the process. Okay, so hyporheic zone. How do we define what is a hyporheic zone? It turns out I didn't know how complex this definition really was and how debated it is. It depends if you talk to a biologist, a geochemist or a hydrogeologist, how you define a hyporheic zone. And the hydrogeologist, I don't know, you all get like really complicated in that definition. There's all sorts of seemingly debates about how to define it. But basically, what I view it as is sort of this region underneath like streams and wetlands, for example, what I'll be talking about today, where there's mixing of surface water, which is usually pretty oxygen rich and depleted in things like nutrients and metals and stuff like that, okay? And it mixes in this area with groundwater coming up, right? And groundwater typically has, very low oxygen or often anoxic, no oxygen, rich in metals, rich in nutrients. So it's carrying it in, and so you have this whole mixing zone in here, and it's more complicated than that. And we're going to be talking a lot about the vertical flux through the hyporheic zone today. But just keep in mind that there's, it's moving, and the water's moving in other directions too, not just up and down. It's moving laterally, and so. But we're going to be talking about this vertical flux today. And what's interesting about what happens in there when you have all this mixing and bringing in of oxygen and anoxic waters and nutrients and nutrient depleted, you set up a really nice oxygen gradient throughout this hyporheic zone, right? So where you would have oxic conditions near the top, you would have anoxic conditions near the bottom, and in this gradient can be pretty complex because water is dynamic, it's moving, it's responding to rainfall events, it's responding to all sorts of things, right? So this gradient is moving potentially up and down in some of these, especially really dynamic streams, and it could be stretching out, it could be getting really narrow. And what happens in that range is you have a complex set of biogeochemical reactions, right? So these are reactions that are happening. You can kind of see what I have this electron or a redox tower right here on the right-hand side. And this is just showing you, if you aren't familiar with redox towers, what you would expect from the surface, like the sediment water interface down through that hyporheic zone, the sets of reactions in order in which you would see them. So for example, oxygen, which is the most thermodynamically favorable electron acceptor, right, would be very quickly reduced by biological processes. Okay, so oxygen can be depleted within the first, millimeters, centimeters. It depends on the hyporheic zone you're looking at. But then eventually move down this tower into less and less favorable electron acceptors. So nitrate, manganese, iron, sulfate down here. I'm going to be talking about the sulfur cycle. And eventually you get to CO2 reduction and produce methane. This is where you get methanogenic production or methane production by microbial processes. So that happens. And why do we care about this? Why do we care about all these biogeochemical reactions other than a genuine interest in biogeochemistry? And it turns out that often these hyporheic zones are referred to as the river's liver. I don't know. I also heard it referred to as the human gut or something. I don't know. It's kind of ridiculous, but I really like the river's liver because a lot of different, all these biogeochemical processes are so important for the health of these systems, especially the health of the surface water above it. Okay, it's where most of the organic matter is buried and where it gets turned over. So important for carbon cycling. All happens in this hyporheic zone. Almost all of it in these systems happens in this, and it can be pretty shallow sometimes, these shallow hyporheic zones. It's important for greenhouse gas emissions, nutrient cycling, nitrate, phosphates. A lot of processing of nitrate and phosphate happens in this hyporheic zone. Contaminants often get sequestered in these hyporheic zones. So overall, it's just really important for water quality, for environmental quality. So as I mentioned, the title of my slide or my talk is something about like underappreciated sulfur. And why would it be underappreciated? It turns out that in freshwater systems, most freshwater systems anyways, sulfur is in pretty low concentration, at least sulfate is in pretty low concentration, right? So if you're moving down this tower, I'm thinking about like in a stream in Minnesota, what would you expect to happen? You would expect oxygen and nitrate reduction. There's so much iron around, right? Iron is so abundant in most systems in Minnesota. So you would expect iron reduction to be this really important process. And then we skip sulfate reduction. I don't know, people say, oh, there's no sulfate around, so it just quickly gets reduced, and then that's the end of the story. And then we move on to methanogenesis. It turns out, and it's not just my group, and I'm going to show you a lot of the evidence for this, that we shouldn't be overlooking the sulfate reduction and sulfur cycling. It actually might be really important, much more important than we anticipated. There's a number of groups who are alluding to this, and a lot of this is spurred from the marine system studies where sulfate is actually quite abundant, but they've worked out some really interesting things happening with sulfate that I'm going to talk about that we think is happening here in our freshwater systems. So why should we care about sulfur in these freshwater systems? Well, yes, sulfate is low in most systems, absolutely true, in most streams, but it is actually increasing. There's a lot of studies. There's a group in Colorado, Evelyn Hinckley. Her group is modeling sulfur and what the expectation is. And it turns out that we're expecting to see increases in sulfate in most of our stream systems. And it has to do with agricultural inputs, a lot of fungicides and a lot of some of the newer sort of treatments that they're using in agriculture are really rich in sulfate. So we're starting to see increases in that. Industrial inputs can add a lot of sulfate. So people familiar here in Minnesota with the controversies happening up in northeastern Minnesota related to iron mining and the sulfate releases. So we have another study on that. It is important, and it is potentially increasing. So I'm gonna walk you slowly through this diagram because I realize it's a really complex diagram, and I'm gonna walk you through it. But what we think so far and what's happening in our systems and what other people have documented, and you can broadly see, I've circled this, I'll define cryptic in a minute, but you see this sulfur cycle, right? So you see, what you can see is that there are oxidized forms of sulfur, right? Sulfate in the lower right. And then you can see it gets reduced and reoxidized. So this is cycling, right? We're reducing something and then we're oxidizing it, right? So remember that these are all involving the transfer of electrons, right? So the cryptic sulfur, well, the sulfur cycle, what we think is happening, and I'm going to explain this more, is linked to the iron cycle, right? So they're coupled reactions. So if you have something like sulfate being reduced, right, to sulfide, and then, so it's now sulfide. Let's put sulfide up here. And we have iron 3 in our system, and they kind of move together, right? They're kind of, I like to think of them as like gears, like cogs, and they move together, and the sulfide gets oxidized, iron 3 gets reduced, right? And there's a transfer of electrons between those two. So we think those are tightly coupled, but you might say, okay, you just showed me this diagram where iron 3 should be totally reduced before sulfate reduction starts happening. Turns out environments are way more complex than that. And I'm not going to show a lot of the evidence today, but there is a lot of evidence in our studies and others that show that actually there's still a fair amount of iron 3 in a lot of environments, even where we expect it to be totally reduced. So iron 3, we can assume, is present in some form, and I can talk about those later. But sulfur is coupled to iron. Interestingly enough, it also might be coupled to a methane cycle. You can see that here, right? And so sulfate, right, is being reduced, and you can see methane on the lower left here, right, is being oxidized. And so I think that is going to be an important process to start studying in the future. I'm not going to talk about that data because we're still generating all that data right now. Sam is working furiously in the lab trying to analyze all our methane data. But I think this is going to be an important part of the story moving forward as well. But we have this sulfur cycle, okay, and it's coupled to other cycles. So why do we call this cryptic? Why, like, what does that even mean? So when I say cryptic, I just mean in hidden, right? It's hard to see, it's hard to observe because sulfate is in low concentration relative to a lot of these processes. And it's, you know, it's just hard to observe something happening that's happening in low concentration. But if you have these processes happening where, let's say you keep bringing sulfate into the system from maybe hyporheic fluxes or other processes, maybe that sulfide gets regenerated and I just have sulfur intermediates. That means intermediate valence, so like elemental sulfur, polysulfide, thiosulfate. This is a group of things that we're just calling sulfur intermediates, right? So you can see that, and it starts to get reoxidized through microbial processes. And so what's spurring that? Well, this coupling to this iron cycle is probably doing that. And so you have this constant regeneration, even in a totally anoxic system, right? It's anoxic, so we shouldn't see oxidation happening, but we do because of this coupling with iron. So you're regenerating more and more sulfate, and it just keeps cycling like this, right? It just keeps going round and round and round, and it can happen at pretty fast rates because these microbes are really efficient at their jobs, right? So you have all this this sulfur cycling, it's being reduced, and then it's reoxidized, and it's reduced. So it might just be that, you know, initially we thought, okay, the original hypothesis going into most of these studies and sort of like the paradigm is that sulfate gets reduced to sulfide. There's a lot of iron 2 around because iron has already been reduced, and it precipitates out this iron monosulfide or pyrite. End of story. It just gets stored as iron sulfide down in the subsurface. It turns out when we actually look in a lot of environments, that's probably not what we're seeing. And I'm going to show you some of our data that supports this. But we do see all of these sulfur intermediates and this reoxidation. So what you're going to see today is all the evidence, microbial evidence, the geochemical evidence that we have that suggests that this is actually quite important in these hyporheic zones and potentially other freshwater environments as well. Okay, so when I say cryptic, it just means like a hidden, fast cycle that's happening. It's just hard to track. Okay, all right, so what are the objectives of this study? Well, again, I mentioned kind of what the overall objectives of hydrobiogeochemistry are, and it's really to seek those connections between, you know, the cryptic sulfur cycle. Here we're really focused on the cryptic sulfur cycle. Whether or not it's connected to iron or methane cycles is also important. But how that's responding because of this hyporheic flux, right? You have, especially in the system I'm going to show you, you have rivers that are going dry, penetration of oxygen going down, then they fill. You see huge changes in the flux, the hyporheic flux and exchange through these hyporheic zones. How is that affecting it? And then once we figure that out, how can we make predictions, for example, going into the future, right? And this is going to be really important, especially if we start thinking about the carbon cycle. We want to know a lot more about what's happening with carbon, with methane emissions, and stuff like that. So we want to be able to develop some models to be more predictive about what this should look like in systems. So that is work going on into the future for predicting water quality. So we're doing a lot of this work. It's called the Argonne Wetland Hydrobiogeochemistry SFA, Science Focus Area. SFAs are something that DOE likes to focus on. They have these like areas where there's all these scientists doing all this work at one area, targeting all these different things. So this is in South Carolina on the Thames branch, which flows into the Savannah River. And this area is interesting because there's a lot of uranium here, a lot of uranium contamination. It turns out this reactor, this M area facility, was enriching for uranium. Fortunately, not plutonium at the time, but they had a spill, and it flowed down. You can see in the left-hand side, it flowed down right down this river into the Thames branch, this little stream into the Thames branch. Fortunately, we were just upstream of that, right? So we were just upstream of all this uranium contamination for a good reason. But also, well, it's just a lot easier to do our work when there's not a ton of uranium around. But also, all the reactions that we're looking at are really important for uranium transport and mobility. So we can take our work, show them what's happening, and they can they can try to figure out if similar things are happening, for example, downstream where the uranium contamination is, so that they can understand these processes with respect to what might influence uranium mobility. So that's part of the bigger project. So we go in the field, we do a lot of different work in the field. You can see it on the left image. Those are some of our surface and groundwater monitoring wells. We're monitoring for the past several years what's been happening there on like a 15-minute basis. So we have a nice long-term record of what's happening there. We also take groundwater we take sediment cores into the hyporheic zone where we extract pore waters, and we look at the sediments. And we do a lot of, you can see there's a list of a whole bunch of analyses that we're doing. I'm not going to show you all this data, okay? But I am going to show you some of the data that we have for this that leads us to believe that this sulfur cycle is quite important. Okay, so we set up, one of the first things we did when we went out there is try to figure out where do you set up these like long-term stations, where in the river. So we went out and we set up a number of different sites. You'll see these images on the top. If you look, these images were taken in January of 2019 when it was fully inundated. And we had very little knowledge of this stream going into our system or into our study. So it was fully inundated. We set up these different areas. On the left-hand side, you'll see this, what we call the stream. And there's what we call no flocks. So in that image with the syringe, you see these orange fluffy stuff. That is what we call flock. These are iron oxide microbial mats that are throughout the wetland. Okay, so we know there's a ton of iron in this system, like an extraordinary amount of iron in this system. It's quite beautiful. If you love iron as much as I do, you go out there and you get very excited. So we have our no stream flock, our no flock stream. We also have, you can see in the middle images, right, what we call the tributary, and then on the right-hand side, you see a set of images, what we refer to as the wetland and sulfide flock. And these are areas where we see all of this orange stuff. Turns out when one of our colleagues went back there a few months later, it was totally dried out, completely dry. Fortunately, we have a record of when it goes dry, and how long it goes dry, and when it inundates again, and what's happening with the flux throughout this stream. It turns out it's really quite sporadic, and it's hard to predict. Like, there's no clear seasonality to this stream. It just kind of does what it wants. It's very interesting, also kind of challenging. So these are the wetland flocks up close. Hopefully I can play this movie from here. Oh, I think I can. Okay, so I got really excited when I saw these. So this is a picture or a little video in the wetland. And you can see it's close up, and you can see how incredibly gooey and mat-like this iron flock is. And I can pan up and you see like these big bulbous, streamy looking things. These are microbes precipitating iron oxide. So they're generating energy, or they're getting energy from the oxidation of iron 2 coming up from the subsurface and precipitating these iron oxides. So they live on this like fringe of oxic and slightly reducing conditions. And it's beautiful. They move, they like break off in chunks and they start to move downstream. So you saw there was flow through this wetland. Yeah, I love those things. I get very excited every time I go out. So, whoops. Okay, so what do we know about the hydrology? And I'm not going to go too much into the hydrogeology, but what I'm showing you are head gradient plots for our different sites, just for a short time period. I didn't want to show like the years and years worth because it gets hard to see what's happening. But these are kind of representative of what generally happens at our sites. And so on the top, you see this like tributary, and in the middle, you see a wetland. Okay, so what we determined is that if you look at the plot of head gradient, so positive is on top, negative head gradient is in the middle through time, okay, what you see is that sometimes you have like this positive, in this case, positive means a gaining stream. So that means upwelling, right? Upwelling of groundwater up into the stream, right? That's what we call a gaining stream. And then it switches, it changes pretty regularly actually between gaining and losing. And then it gets dry for a while and it fills up. So we see this pretty regularly. And this one also more strongly gaining. This stream, the regular stream that doesn't have the flocks, is typically losing. So you can see it has like a negative head gradient here pretty much the whole time, which means it's losing. So the surface water is flowing downward. It's downwelling into the hyporheic zone. And this is consistent. So anytime we see flocks, anytime we see these microbial flocks or microbial mats, that means, that indicates to us, it's very visual, a gaining upwelling stream system. Okay, so it's very easy for us to track. We take samples throughout at different time periods, different seasons, and I'm going to show you some of the data. I'm not going to show you any, but what I really want you to know is that I'm going to tell you right up front what we're seeing. There's time, right? There's temporal trends. There's spatial trends, because we have all of these little subsites located in a pretty small area, actually. And the geochemistry, as I'm going to show you next, really suggests an anoxic oxidation of sulfide. Right? So you have this reduced, sulfate's been reduced to sulfide in the hyporheic zone. And what we're seeing, and I'm going to show you, is that there's oxidation of the sulfide, even in anoxic conditions. Okay? We see this through the presence of a lot of intermediate valence sulfur phases, and I'll show you how we get at that data. And we think this is likely coupled to iron and carbon. I think that said sulfur. Okay, so what is our evidence? Well, we have some pore water evidence, and this is just some of the data. And what you see is just two different time points, and I'm showing you January on the left, August on the right, of aqueous sulfate in the surface water and the pore water. So what you see are these profiles of depth. This dashed line is the sediment water interface. So those points above it are surface water. The points below it are pore waters in the hyporheic zone. So pore waters are just those interstitial waters between all the sediments, right? And concentration of sulfate on the x-axis going from 0. And in this case, you see it only goes up to 20 micromolar sulfate. So what's happening in January? Not much. Not much exciting is happening. There's just not much happening, right? You see concentrations of sulfate in the surface water, 10 to 20 micromolar. It really doesn't change with depth. It doesn't matter which system you look at. There's just really not much. It's cold. Maybe the microbes are really not very active when it's this cold. We know it's been inundated for a long period of time, or at least months going before that. When we went back in August, it's warm, things are really active, microbes are very active. Also, it was really interesting when we went back in that August because it had been dry for about a month. When it's dry, oxygen is going to diffuse down into the hyporheic zone, totally change that redox gradient, right? Totally alter that redox. Things that were reduced are now oxidized, or at least some of them are. And then it just, right before we went, thankfully, it filled up so we could take samples. And what we saw was really intriguing, and we're still trying to wrap our heads around this, to be honest, is that sulfate, again, was pretty low. But if you look at this range on the x-axis of the concentrations, much bigger range, right? We see much higher concentrations up over 500 micromolar sulfate from about 0 to about 10 0 to 10 centimeters depth. That's a lot of sulfate at depth. Why is that so much sulfate there? I'm not sure, but it's really high. And you can kind of see these red, orangish colors, right, on this right-hand graph. So those are our upwelling, our flock systems. That's where it's really, really high. And the blue is the stream, this downwelling stream. Sulfate's still pretty low. But even if sulfate's low, it is never, ever 0. And that's an important thing to reconcile, because why does sulfate never go to 0? It's actually hard for the modelers, it turns out. So for Crystal and Sam, who are modeling all this data, it's actually hard for them to get to a level of sulfate that we actually see in the environment. Most of the modeling work that's done would deplete sulfate immediately, and it would be 0, it'd be gone, and then you'd see like methanogenesis happening. Turns out, no. So it suggests that there potentially is a source or regeneration of sulfate at depth. So that's one area of evidence. The other thing that we looked at is what's happening in the actual solid phase. So we took our cores and we sectioned them with depth, and then we brought them to the synchrotron. where we do something called X-ray absorption near edge spectroscopy. This is great technique when you have really complex media and maybe not as much sulfur as, say, some of your other elements. You can tune the energy of the photons coming in, and you can tune it to look at sulfur, just sulfur. What is all the sulfur? And tell me, is it reduced sulfur? Is it intermediate valence? Is it S0? Is it S4? Is it S6? So this is the technique we use to help us get at really what is in the subsurface. So what I'm showing you is just one month, right, August. And I will say that we have a lot of other data points. This is all pretty consistent across no matter when we sample. And so again, you see these plots for versus depth. So 0 to about 20 centimeters down, right? And you see all these different colors, and I grouped them for you. And I'm showing the relative abundance, so the percent relative abundance of just the sulfur pool, right? This isn't like the whole sediment. This is like just the sulfur pool in the sediment. So what we can say is that, yes, there is reduced sulfur. There's iron disulfide and iron monosulfide are actually grouped together in this gray. Turns out there's really not much there in terms of this inorganic iron monosulfide, which people would have predicted to be the primary product of sulfate reduction. What we see is a lot of what we call intermediate sulfur. And I have these grouped together just out of convenience because there's a lot of different references. And what we see is that, for example, elemental sulfur, that sort of bright yellow color, and then organic intermediate sulfur and so on is there these like yellowish colors. We see a lot of that, a lot more than we really kind of expected. And then we do see a fair amount of oxidized sulfur at depth, okay? So those are those bluish colors. So we don't see obvious trends with depth, and we don't see it temporally with this, as we did with the aqueous phase, okay? So that's one interesting observation, the amount of intermediate sulfur. And I should say that with the elemental sulfur and with the inorganic, there's a lot of different phases. There's thiosulfate, tetrathionate, there's polysulfides, there's a whole suite of things wrapped into there. But they generally fall in the range of sulfur 0, or with like sulfur 0 to like +4 or something like that. Okay, on average. So not only do we see something, all these intermediate phases, we saw something else that kind of surprised us. But it's a trend that we're starting to see in more and more systems that we look at. We would have expected most of the sulfur to be in an inorganic phase, but it doesn't seem to be the case. And in fact, almost most of the sulfur are things that I have highlighted here are these organic sulfur phases. So when we look at these hyporheic zones, most of it is organic. There are clearly inorganic phases, but there's a lot of organic sulfur. We're still trying to wrap our heads around what all of this means in terms of the sulfur cycle. Is organic sulfur, are these compounds available to microbial reactions? We don't really know that. Are they reactive? Do they oxidize and reduce? Are they stable? We're still trying to learn about, there's a lot of questions about organic sulfur in the environment and what it represents. Some work to be continued for sure. So, okay, so we have geochemical evidence. We have evidence in the pore water, we have evidence in the solid phase, but what about the microbial evidence, right? A lot of these reactions are not abiotic. They're created, they happen because of microbial actions. So what are those microbial interactions? And how does that relate to sulfur cycling? So I'm gonna show you just some of the work that Shreya has been working on. But this is what we use quantitative functional sulfur gene approaches, basically. So we're only looking for, in this case that I'm going to show you, sulfur genes, genes related to sulfur cycling. But we do think we see a lot of evidence of an active cryptic sulfur cycle here using this evidence as well. And so this is a technique called quantitative PCR, where it's really pretty cost effective if you know what genes you're looking for. Unfortunately, with the sulfur cycle, we actually know a lot of the genes involved with sulfur cycling. So we can actually extract all the DNA from our core sediments, and probe it for just, for example, I have 3 genes listed here. Shreya's done a couple more, but we have a couple genes involved purely in sulfate reduction, right? You see these 2 genes here, this APRA and DSRA, gene names we're not going to worry about, but they're involved in sulfate reduction, right? They take sulfate all the way to sulfide. So, and then on the right, we wanted to look for some genes that would give us some indication about sulfur oxidation. So we're using one gene called the SOX gene, in this case, which is a process, an enzyme that can take thiosulfate to elemental sulfur or to sulfate. Okay, so we're going to quantify these genes in the environment, and the nice thing is we can look at a lot of samples very cost effectively. Okay, and I'm just showing you a really small segment of this data. But if we look at the sulfate reduction genes, right, what we see is a plot, again, let's see, here we go. Depth in 1 month, right? So they're plotted versus depth. And they have, again, we have the same color scheme. So our, flock generating systems, those are in like red, orange, and yellow. Our downwelling stream that has no flocks is in blue. Okay. One of the things that we see is that sulfate reduction in the gaining stream systems is more active than in the losing stream systems. Okay, so it's more active in these gaining systems. Still trying to understand exactly what's causing that. Probably because the amount of sulfate that I showed you, at least in August, right? There was a lot of sulfate, so it makes sense that there's a lot more sulfate reduction happening. And then there's also a lot of sulfur oxidation happening. So this is the SOXB. So this is the relative abundance. Sorry, I should have mentioned that. This is the relative abundance of the SOX gene. And you can see how many copies. So this is like over 1 million copies of this gene are present at depth. So there's evidence that there's sulfur oxidation, microbes that are capable of sulfur oxidation, way more abundant in the subsurface than they are in the surface, which is kind of intriguing. And we think it has something to do with the hydrogeology. We're trying to kind of wrap our heads around that. So yes, we definitely see evidence of sulfur oxidation. Oh, interesting. Oh, there we go. Okay, so Shreya also used something called shotgun metagenomics. This is where we take all the DNA and sequence all of the DNA and then try to make sense of it. How is it, Shreya? It's fun. Right? It's computationally very intensive, and it's also quite expensive to do. So this is why we don't do all metagenomics all the time, but we actually ended up doing a lot more metagenomics than we anticipated because it was so interesting. So we're looking at all the genes. So I'm going to show you sulfur, but there's, we have information about nitrogen cycling. We have information about carbon cycling. We have information about all those biogeochemical processes happening, but I'm going to focus on sulfur. So I've been I've been showing you these like really reduced spur cycling forms, but it turns out it's more complex than that. You can see this is a diagram showing all the different sulfur cycling pathways for inorganic sulfur. We're trying to wrap our heads around organic sulfur because there's a ton of organic sulfur cycling going on in these systems. Those genes get even more complex, but these are just the generalized pathways in this sort of like reaction network of, the reduction of sulfate to sulfide, then maybe you have these like oxidation and disproportionation. There's all these different reactions happening. So I'm going to simplify it. Most of these sulfur intermediate forms are circled in that blue circle. Okay. And these transformations, if they're present in a system, are red. I'm not going to get it's hard to show that, but basically we have most pathways represented: sulfate reduction, sulfide oxidation, sulfur oxidation, thiosulfate disproportionation, and thiosulfate oxidation. So a lot of these things are happening in the subsurface all at the same time, but that doesn't give us an indication of how abundant or how prevalent these things might be. So here what we're showing is a heat map of which are the most important processes. And what we think here, these are all the different processes that you saw on that previous map on the x-axis, right? And you can sort of see this scale of relative abundance from, white is low relative abundance to these dark blues are really high of relative abundance. And one of the things I wanted to show you is you see this like, I've highlighted a couple of processes. Sulfur oxidation is like highlighted in this like bright blue, and sulfur reduction is in the orange. And what you can see overall is that sulfur oxidation genes are way more abundant and way more important throughout the entire system. This is the tributary, this is the wetland, the sulfide pond, this is all the systems. Doesn't matter what system we look at or what depth they are. These processes are incredibly important, the sulfur oxidation, no matter where we look, okay? One of the thing that's highlighted in red is dissimilatory thiosulfate oxidation. That's one of the most dominant processes that we see in the subsurface. So we're gonna try to figure out more about this. Shreya just plotted these up for me the other day. So, okay, so what does this all mean? Let me wrap it up. Do we see evidence of a cryptic sulfur cycle? Have we convinced ourselves? Yes, we've absolutely convinced ourselves. We see this in the aqueous phase geochemistry. We see it in the solid phase geochemistry. We see this in the microbiology, right? A lot of these reactions are microbial. We see it there. Yes, I'm not showing you the iron data. It's just, we have a lot of data. Not showing you it all. So I wanted to be selective. So the sulfur, yes, is it happening? I'm pretty convinced it's happening. How does it respond to, I wanted to bring it back to the hydrobiogeochemistry. How does it respond to hyporheic flux? That is a very challenging thing to answer, it turns out. So Sam and Crystal are developing these reactive transport models to incorporate all of these sulfur reactions. Those sulfur reactions are not typically found in most reactive transport models. I can't think of an instance where people have really done a job of incorporating these. And not only that, we have to figure out which ones are the most important because they can't model everything, right? They can't put all those reactions in. Which ones are the most important? So now we have some constraints based on the microbiology, geochemistry, what they can really focus on. So they can sort of bring this around so we can better understand, like when it goes dry, when these systems go dry, what would we expect? What should happen with all of these biogeochemical cycles? When it fills up and it's inundated for a long period of time, what happens? What happens when it's like moving up and down all the time? So I think there's a lot of questions to answer still. These are things we're all working on. Is this unique? I mentioned this before. No, it's not unique. We published a study up in northern Minnesota. Josh Torgeson, some of you remember, this was part of his thesis. Observing similar things, but in a sulfate impacted mine system. So I want to thank you for your attention. I'm happy to take questions. I don't know how to do that on, hopefully something will happen on if people have questions online, but yeah. I do remember you. Hi.
10 years ago, we did a lot of studies on the sulfur and oxic isotopes of sulfate in the St. Louis River. And I don't know if you looked carefully at that data, but what we found was that the oxygen isotope ratio of the dissolved sulfate was one value in the mine, and it slowly changed all the way downstream until you got to the estuary on Lake Superior, and the oxygen isotope value was different. Did you ever look at that data and try to explain this?
Yeah, we have looked a little bit into that data, and we thought about doing some isotope work, at least for our system in northern Minnesota. I think we were having problems getting enough sulfur to actually do that. I can't remember exactly why we abandoned ship. Maybe it had to do with COVID. I don't know. But yeah, I do. I'm not sure. So does that indicate mixing, or does it indicate something totally happening in the break zone? Yeah, so you've ruled out mixing, so it must mean that something is happening in the hyporheic zone to change these values. Is that what you're suggesting?
Well, all I know is that the river water, the sulfate comes in that river water, to change its oxygen isotope ratio without sulfur isotope ratio. So suggesting the sulfur was staying in the water, but the oxygen was exchanged. And it was beyond what we could explain with just mixing with water coming in from the sides. So I'm just gonna.
Yeah.
I was really anxious to somehow interpret it.
Yeah. Yeah, this model didn't exist when we were doing sulfur, some of the sulfur isotopes. Some of the, I don't know if people can hear online, but there was a question about some of the earlier work. I guess, were you with the DNR then? Yeah, with the DNR to look at sulfur isotopes up in northern Minnesota and sulfur oxygen isotopes. But yeah, maybe we can revisit that and see how this explanation might influence the interpretation of some of those isotopes. But, yes, yeah. Right, right. Yeah, and Megan did her PhD here, didn't she? Or graduate something? Yeah, okay. Thanks. Thanks for the question. Does anyone else have any questions? Bill? Yeah, so there's a question about what are these organic sulfur species? So when we do the sulfur veins, we can get a decent understanding of them, but it's not great. We need to switch, now that we know how much organic sulfur is in there, if we want to really define what they are, we're going to have to start using other techniques. It's challenging to identify those organic sulfur phases. And so we're going to have to think about how badly do we want to know what exactly they are and what they might do in terms of their reactivity. I think there's a ton of, there are a lot of questions about what is all this organic sulfur? Is it reactive, right? Can it be reoxidized? Can it be reduced? Is this just like a sink for sulfur in systems like this? I don't have a very good answer to that. I think there's a lot more research that needs to be done to sort of figure that out, not just, you know, in our group, but like more broadly speaking and for the field. I think there's a lot of interest in improving our understanding of organic sulfur and what it contributes to organic, to sort of sulfur cycling in really any environment. I think we're coming to a consensus that it's been overlooked and that we don't really understand too much about it. I have an idea of, like, you know, some of them are like, I think we should also be looking at things like polysulfides, some of the organic polysulfides. When we do our sulfur fitting for our Zanes data, it suggests that there's a fair amount of polysulfide in there, and polysulfides can be inorganic, or they can be like long chain inorganic, or they can be cyclical organic phases. And I think we need to start looking into that a little more. We need to improve our reference library, but even with that, I think we just need to switch to different techniques to really. Yep. So in the aqueous phase polysulfide, so we haven't measured polysulfide, and that's one of the things now that we know so much and have looked at this data a little better. I think we do want to look for polysulfide in the aqueous phase as well. So at this site, Marcial Telefair is actually working there now, and he's been working at a slightly different area, but I think we're meeting next week to potentially, he uses electrochemical techniques and can actually measure polysulfide in situ at the time. And so maybe working more closely with him to use a combination of our techniques and his techniques to really kind of, you know, flesh out these weird sulfur. They're not easy to measure, right? They're kind of enigmatic. If you don't preserve them properly, you know, you got to measure them right away. So his approach of the electrochemistry might be a really good approach for getting at that for the aqueous phase. Yeah, questions from students. I forgot. I've been on sabbatical for a year. I forget these things. Students first, yes.
You tried like using the metagenomic data to assemble metagenomes or consider doing 16S, 18S?
So we do have 16S data as well. And I didn't show that here. It's a lot more data to show. So we know that the metagenome assembled genomes, Trey and I have been talking about that, which to target. Do we do all of it? What do we really, do we focus our attention on a certain things, parts of the story that we're really interested in? I think it's going to be important, especially for thinking about, okay, yes, there is iron there, but what's oxidizing the sulfide? Like what's the terminal electron acceptor? Or is there some other, people have proposed cryptic nitrogen cycling in these systems. So is there a coupling with the cryptic nitrogen cycling and the sulfur cycling? And you would have to get at it with using some of these techniques. So I think that's something to resolve for sure, and an approach that we'll start taking. But yeah, that's a good question. The 16S data isn't as exciting as we thought. There's, there are subtle differences in the microbial community, but they're not like, there's nothing jumping out at us that's like really important in their trends. Yeah, thanks. Chris?
Yeah, I was curious, it looked like a lot of the sites, the total organic sulfur pool seemed pretty consistent with that, but do you see changes in the constituent species from one to another?
Like within the organic sulfur pool, if you mean? We do have that data, and we have looked at that data. And the reason we grouped it is because we just don't see a lot of differences in like what we're saying is the organic sulfur pool. Like, I can't remember what, we have a number of different species, different references that we use. And yeah, I'm not convinced we see any real clear pattern there. Maybe subtle differences, but with this technique, I wouldn't like say that, it's a big enough change to be confident that there's a major trend to observe. But yeah, we have looked at individuals instead of grouped.
Microenvironments in the flocks that are acidic, and that contributes to
Microflocks, are microflocks in the flocks? I mean, microenvironments in the flocks or in the sediment in the hyporheic zone? The flocks themselves, because those are sitting way above. So we haven't actually measured the flocks. So Clara Chan at University of Delaware is looking into the flocks. That's like, those are the flocks that are in the surface water. But so we haven't looked at what's happening in the flock system. Clara just started working on that. So I don't really know. Why are you asking? Oh. So we don't actually see, we measure the pH. We don't see it that acidic. And I can't remember if we stuck our probes into the, right into the flock. I think we were just mostly staying away from the flock. So I'm not sure I know off the top of my head what the pH looks like right in those flocks and whether they're acidic. But generally the pH is fairly neutral. I can't remember exactly what it is. Trey, do you remember? Can't remember. But it's nothing like real acidic by any means. But I think it's interesting. Those flocks are interesting, right? Because when those systems go dry, they draw down all of this. Like you saw those like goopy, like iron-rich microbial mats. What happens to them? They're not moving downstream fast enough to make any sort of difference. They're like being sucked right into the subsurface. And we actually see when we went back there after it was dry and we measured iron levels in the subsurface. So it went dry and then we went back like a week later and it had been inundated again. The iron was, I can't remember the concentration, but it was like outrageously high. I've never even measured iron so high in an environment before. It was really, really high. So that clearly those microbial mats and all that organic matter are sinking down into these hyporheic zones when they go dry. Potential sources of sulfur, nitrogen. So quite interesting. Yeah. Yes.
Whenever you saw that the water in this video, it looked pretty thick.
Yeah.
It didn't just look like
Yeah.
Or were there other things that they around it?
Yeah, that's a good question. So the microbial mats are really thick in that wetland environment, but they're not so thick that like when you step into it, because we often have to go into it to sample and we disrupt it, and it clears out around that area very quickly and for days. And then maybe if we go back at the end of the week, we start to see that they're starting to reform there again. So they reform very quickly. But I don't think it's like, there's definitely water flow through those systems, through those flocks. We can see it, we can measure it. So are they choking things per se? I don't think so. There's not a lot really living in that stream. We try to avoid all the spiders and snakes. There's water moccasins in the area. I don't know, doesn't make me happy. But you know, we don't like, it's so small and it just like it goes dry so often. There's just not a lot living there. We do see trees and other things, some plants, but you know, there's just not a lot of life in those, like the wetland and the stream system. Yeah, good question. Okay, I think there are some people online. I don't know. Oh wait, I do see something in the chat. Maybe I can. Oh, okay. There's no questions. Great. It's 5 o'clock. Thanks. Thanks, everyone. Hi to the online people. Thank you. All right.